Now we will continue with the primary organ. The second primary organ is thymus. So, thymus is the site of T cell development and maturation. And as an organ, this is a small bilobed organ located just above the heart, right? And these are it's a bilobed organ, and each lobe is surrounded by a capsule, right? And these lobes they are internally divided into lobules, right? And this lobule division is made possible by an internal growth of the capsule and other structures, and these are called or there is a strand of connective tissue to separate these structures into lobules, and these strands of connective tissues are called trabeculae, right? When we see the image, you will be much clearer about that. And when we take a cross section of the thymus, you can see two parts, an outer cortex region and an inner medullary region. And the outer cortex is much populated with rapidly dividing immune cells. That means rapidly dividing cells in the thymus. They are actually those cells, those immune cells which are maturating in the thymus are called the thymocytes. So there will be rapidly dividing thymocytes in the cortex, whereas medulla is having fewer number but mostly mature thymocytes, right? And not only as in the bone marrow, we have seen that there are various other cells, right? Similarly, in thymus also there are various other cells which are also significant in the development of the T cells. So these non-lymphocytic cells which are significant in the T cells are that include epithelial cells, dendritic cells, and other macrophages. So we are going to see which are these cells and what are their roles in the next segments. So this is the image that give you an idea about the cross section of thymus. So here you can see the medullary portion, the cortex region, and also you can see the trabeculae, which is separating the lobule. You can see the capsule. Then there we have the thymocytes. The thymocytes are, there are more number of thymocytes in the outer cortex region. But when you come to the medullary inner portion, there are a small number of thymocytes are found, but most of them are in the activated stage or in the active stage, right? And there are some other cells also given with certain names that we will see. These are the non-lymphocytic cells which are but significant. So one of the named non-lymphocytic cell present in the outer cortex of this thymus is called the nerve cell, right? So this nerve cell is nothing but an epithelial cell with long membrane extension. That what actually they do is they will keep or they'll surround a large number of thymocytes, like around 50 to 200 small thymocytes, which are actually in the developing stage, that they are in the dividing stage, will be hold together by this long extension of the membrane of these nerve cells. So nerve cell is nothing but an epithelial cell, but they have long extension, and this extension is supposed to cover around 50 to 200 different thymocytes, right? So in short, that will become a multicellular complex, right? And it is also suggested that there are certain functions like arrangement of T cell receptor also occurs at these nerve cells. Right. It was believed that this is the site where the T cells are providing some basic information, some basic education is being given for the T cells at the nerve cell, but that is not evidentially supported. But it is supported that there are certain arrangement of T cell receptor occurs at this particular site, right? Now the second one is the cortical epithelial cell, and also there are medullary epithelial cells. Right, these are the normal cells of the thymus. One is in the cortex region, the other one is the medulla portion. Right, they are actually the normal cells. They are not much to discuss on that. Right, then the next one is the Hassel's corpuscles. Hassel corpuscles are the concentric layers of degenerating epithelial cells usually found in the medulla portion, in the inner portion. Right. These are the concentric layers of epithelial cells. The exact function is not clear, but it is believed that they might be involved in the secretion of thymic hormones, because the development of thymocyte is basically is dependent on the presence of various hormones and other chemicals. So the secretion of hormones and other secretory elements like interleukins and other chemicals will be provided by these kind of collection of cells, right? So Hassel corpuscles are degenerating concentric layer of epithelial cells found in the medulla. They are not associated directly with with any functions of the lymphocytes, but it is believed that they may have a role in the secretion of thymic hormones. So now we are moving into the secondary organs. So secondary organs, as already stated, are the sites where the mature lymphocytes are residing. Not only residing, this is the site where they interact with the foreign material. So when a bacteria or when an antigen entered the body, they enter the blood or some other tissues. So these antigens will be entrapped by the secondary organs, right? So within the secondary organ, you need to provide all the necessary equipments in order to combat these antigens. So these necessary equipments of the immune system are nothing but the immune cells. So as already discussed, the immune cells are synthesized in the bone marrow. Then after synthesis in the bone marrow, they will undergo certain maturation process there itself. And these mature lymphocytes, and not only lymphocytes, other cells like the granulocytes and other dendritic cells and all those cells will release into the blood circulation. And from the blood circulation, these cells will migrate into the specialized organs, which are what we call the secondary organs. So these secondary organs are actually collection of mature lymphocytes and other immune cells. They are ready to attack a foreign substance if they enter that particular site. Clear? So how do they perform their function? That's what we are going to see. So here we have listed spleen and lymph node are the organized secondary organs in the immune system. And in short, spleen is responsible for the elimination of antigens which are entering the blood circulation, because blood is the major body fluid, and blood is the site where easily the foreign material or the antigens will enter, right? If you have a wound, or if you have a tissue eruption, mostly the foreign material will enter into the blood circulation there. Then from the blood circulation, they enter various other parts of the body. And the condition by which the blood is being populated by foreign bacteria or foreign antigens, and that leading to certain condition, is called the bacteremia, right? So the presence of bacteria in blood is called bacteremia. So in all those situations, what actually they do is, these bacteria will be trapped in the spleen, right? So spleen is responsible for this elimination of any antigens, not only bacteria, any antigens which are entering the blood circulation. Whereas lymph node, on the other hand, is working to filter the lymph, right? Lymph, I'm not discussing again, this is the body fluid which actually coming out of the blood, and that will be collected back into the blood. So before recollecting this lymph into the blood circulation, they are filtered by an organ called lymph node, right? So let's see the secondary organs. First we will see about a spleen. So spleen is the major organ which filter blood. This is the secondary organ which is located in the left abdominal cavity, in the upper position of the left abdominal cavity, right? And just like in thymus, here this organ is also surrounded by capsule. There are tuberculae to divide them into lobules, and also there is the network of tissues which are maintaining or which are supporting the morphological structure of the spleen are called the reticulum. Right. And similarly, the blood vessels which are entering and exiting from this organ is through a particular point, a particular invagination called the hilum. Right. So there are arteries and veins coming and leaving these organs through this hilum. Right. And when we see the anatomy or the cross-sectional view of the spleen, you can see there are certain regions called the red pulp, which is the exterior portion, and the white pulp, which is the interior portion. Right. So there are red pulp in the exterior part and white pulp in the interior part. Right. And you can also see the capsule. There are trabeculae. There are veins and arteries which are entering through the hilum. And this is the structure you have to remember, you have to keep in your mind. Now we are going to discuss all these parts separately. So the red pulp is the actually the largest compartment, and this is the exterior portion. Right. And the most significant thing is that there are numerous venous sinuses situated between arteries and veins. You know what are sinuses? Are nothing but this is the junction between the arteries and veins. Right. These are the junction between arteries and veins. All right. So usually blood is filtered through these sinuses. So these sinuses are responsible for the filtration of blood. Right. And during this filtration, what actually happens is that all the foreign materials like bacteria and other antigens, and also the senescent immune cells like the aged RBCs and WBCs also will be eliminated during this process of filtration. And for that, these sinuses are populated by a number of immune cells like macrophages. They'll be involved in phagocytosing all these cells. And not only these phagocytes, not only macrophages, some other cells are also associated with these functions like neutrophils, eosinophils, then other lymphocytes like plasma cells also present in the red pulp. So this is a major site where the filtration occurs. Right. Now moving into the white pulp. So this is actually the internal portion. This is the site or the lymphoid tissues which are surrounding the branches of the splenic artery. Arteries are the blood vessels which are coming into this spleen. So covering these organs, covering these vessels, there are a layer of lymphoid tissues, and which actually the immediate layer of tissues covering this region is called periarteriolar lymphoid sheath or PALS. And this PALS is rich in T lymphocyte. Right. This is highly populated with T lymphocyte. All right. So the T lymphocytes are actually, we have to remember that T cells are synthesized in the bone marrow. They are maturated in the thymus, and from there they release into the blood, and from there these mature thymus or mature T cells will be migrated into the spleen. Right. So these are rich in T lymphocytes. So not only T lymphocytes, B cells are also present. Right. So within the PALS, there are certain regions, certain concentric regions where the B cells are concentrated. And these regions are called follicles, or collectively these are called Malpighian corpuscles. Right. And there are two types of such follicles called primary follicles and secondary follicles. The primary follicles are the B cell, both are B cell predominant regions, but primary follicles are having only a central mantle zone, and there is no germinal center. Right. A germinal center is the site where the B cells are actively dividing. Right, whereas mantle zone is rich in resting B cells, which express the B cell receptor and the specialized receptor like CD23. So a primary follicle doesn't have a germinal center, but they are having only the collection of resting B cells, which is what we call the mantle zone. All right. At the same time, secondary follicles will have the germinal center and also the mantle zone. So the germinal centers are the area where the B cells are actively dividing and also undergo class switching. Right, class switching we will discuss in the coming session. Is the process by which the different antibodies are synthesized in response to specific antigens, and also there will be affinity maturation through somatic mutations, and also development of memory B cells. Activation is basically T cell mediated, because these memory B cells and other B cell activation will be mainly T cell mediated. Right, because there are surrounding a collection of T cells all around this white pulp region. Right, so that's about this cross-sectional discussion of the spleen. So moreover, you can also see another region called marginal zone in the interface between the white pulp and the red pulp. Right, and there also you can see the T cell population. That is the major population. T cells are the major population, but there are certain B cells also, which are called marginal zone B cells. But these B cells are slightly different from other B cells in the spleen. That means in the follicular B cells, right? So actually, these marginal zone is the first site where this foreign material will be checked by this immune system, or the checked by these immune cells like T cells and B cells. Okay.